So before, I want to kind of start off with a general statement of review on these concepts before we get into those questions on the worksheet. So just in terms of these two concepts, mitosis and meiosis, let's differentiate them. What's the difference between mitosis and meiosis in terms of what kind of cells are made, how many cells are made, and what is the chromosome number for those cells? Okay? You guys know that? So for mitosis, what happens? How many cells get made? 2 cells, right? One cell copies its DNA and divides in half to produce 2 cells.
So it's 1 diploid to 2 diploids?
1 diploid cell to 2 diploid cells. So what does that word diploid mean? What does that refer to? What is the word diploid? Go ahead, Dorian. What did you say? The full, well, it means the total number of chromosomes in a normal body cell, which in humans is 46. That's correct. So mitosis produces cells with a diploid number of chromosomes. And do you guys know what symbology is used for diploid? 2N. That's right. So far we've said that mitosis makes 2 diploid cells. Okay? And what about them? Are they genetically unique and different from each other, or are they identical? They're identical. They're identical clone cells.
You're eukaryotic?
Well, mitosis can be used to make both prokaryotic and eukaryotic cells. Okay? But the type of cells specifically that's made the difference is, are they somatic cells, which are just general body cells, or are they sex cells? So that's the last thing I'm looking for here. Well, what kind of cells does mitosis make?
Somatic.
Somatic cells, regular body cells. Okay. So mitosis makes 2 identical diploid cells, right? They are somatic cells, regular body cells, like your brain cells and your skin cells, and the cell that makes up your lung tissue, and the tissue of your intestines, everything except for one specific kind of cells. All right? So that brings us to the process of meiosis. So how many cells are made by meiosis? 4 cells. And what's their chromosome number? Is it diploid or haploid? Haploid. And what symbology do you use to represent haploid cells?
N.
Just N. And are they identical or are they unique? They are unique. And what kind of cells are made by meiosis? Sex cells. And what's a fancy way of saying sex cells?
Gametes.
Gametes. Good. And what are gametes?
Egg and
Sperm and eggs. Eggs and sperm and eggs are gametes. Okay. So I can add that here. Sperm and eggs. Okay. So that was good news. We got more people here who can actually start going through the worksheet. So does everybody here have the worksheets? This one, the big chapter 8, 9, and 10 review for those who just came in. You guys have that? All right, so let's go ahead and go through this then. So for number 1, if an intestinal cell, what kind of cell is an intestinal cell? It's a somatic cell, right? If an intestinal cell in a grasshopper contains 24 chromosomes, a grasshopper sperm cell contains how many chromosomes? 12, that's right? Half of the number of the typical somatic cell, right? So 12 chromosomes for a grasshopper sperm cell. Does that make sense, guys, right? The diploid number, as given here on the worksheet, was 24. And so the haploid number is half of 24, so that's 12. Okay. All right, number 2, which phase of mitosis is essentially the opposite of prophase in terms of nuclear changes?
telophase?
That's right, telophase. Telophase. Let's think about why.
Is that because when the nuclear envelope reforms?
Okay, so yeah, we can even list out the reasons. Why not? So what are some of the reasons? Well, in the telophase, the nuclear envelope reforms. And what did it do in prophase? What did the nuclear envelope do in prophase? It broke down. Right, it broke down and started to disappear. What else happens in telophase?
The spindle disappears.
The spindle disappears in telophase, that's right. And what happened to the spindle in prophase? It formed, right? And what's the other thing we haven't mentioned yet? What about the chromosomes? What do they look like?
Long.
Yeah, that's right. The chromosomes are uncoiling and elongating in telophase. Okay, so chromosomes uncoil and elongate. What do they do in prophase? What did they do in prophase? They coiled and shortened. Okay? So definitely telophase is the opposite of prophase in terms of nuclear changes because of these three main events that happen during prophase. Number 3, the multiple choice question. So which of the following is not a function of mitosis in humans? Let me read them first so you guys can read the answer. So letter A, repair of wounds. Letter B, growth. Letter C, production of gametes from diploid cells. Letter D, replacement of lost or damaged cells. And letter E, multiplication of somatic cells.
C?
Definitely letter C, right? Letter C, production of gametes from diploid cells only happens during what process? Meiosis. Meiosis. So that can't be something that relates to mitosis. So yeah, definitely choice C for number 3. Because gametes are made by meiosis, not mitosis. Okay? We good right now? Okay. So number 4. During anaphase of mitosis, what separates? Remember? Sister chromatids separate. During anaphase of mitosis. Okay, number 5. During anaphase 1 of meiosis 1, what separates? Homologous pairs of chromosomes separate. That tetrad, remember the tetrad that forms during prophase 1? Tetrad, 1, 2, 3, 4 sister chromatids. Okay? Well, what's going on is that tetrad, the homologous pairs, are separating moving to opposite poles. 1. Okay? And then what finally happens in anaphase 2 of meiosis 2? What's going to separate now? Now those sister chromatids in each of those homologous pairs are going to separate. So sister chromatids separate.
So it's very similar to anaphase of mitosis. That also,
It is.
Yeah.
Yes, everything that happens in meiosis 2 is the same that happened in regular mitosis. It's just that it happens in 2 cells at the same time instead of just 1. Everything unique to meiosis happens in meiosis 1. That's an important thing to remember. Everything unique to meiosis happens during meiosis 1. Okay? All right, number 7, vocab term. So when this happens, when this occurs, homologous chromosomes or sister chromatids fail to separate, resulting in abnormal chromosome number in the cell. What's that called?
Nondisjunction.
That's right, nondisjunction. And just as an extra question here, what's the most common human chromosomal abnormality that happens because of this nondisjunction? Remember that? Down syndrome, right. Down syndrome, which results from trisomy 21. Getting 3 copies of chromosome 21 because of the failure of the homologous pairs to separate or the sister chromatids to separate during the anaphases. Very good. Okay, so how about number 8? Let's get some room here. So you guys got these notes down. These are in so number 8, true or false? Crossing over occurs during prophase 2 of meiosis 2.
It's false.
That's false. What's the correct statement?
It's false.
Would you just go ahead and say that louder? That's right. That's right. Crossing over occurs in prophase 1 of meiosis 1. Okay? So the statement as it was written was false because crossing over occurs during prophase 1 of meiosis 1. Alright, number 9. True or false? Meiosis 2 is preceded by interphase. False. Good. What's the correct statement?
Anaphase 1?
Meiosis 2 is preceded by meiosis 1.
Okay. So you could say it a couple ways. You could say, as Dory said it, meiosis 2 is preceded by meiosis 1. That works. Okay? Or you could even kind of say it differently and say, interphase precedes meiosis 1 only, and not meiosis 2. But I like the way that Dory said it better, actually. Kind of follows the pattern better. So meiosis 2 is preceded by meiosis 1. The whole point to this is that there is no interphase in between meiosis 1 and 2. Okay? There's interphase, and then there's meiosis 1, and then there's meiosis 2. There's no interphase between meiosis 1 and 2. Okay? And then number 10, we had already done that to start things off, didn't we? Okay? So let's just summarize that one more time. So the difference between the end results of mitosis and meiosis, right? So mitosis makes
Two cells.
okay, so mitosis makes 2 what kind of cells? Okay, identical and diploid 2. Identical. Diploid. Comma, somatic cells. And meiosis makes 4 unique haploid. Let's just say sex cells. Okay, we won't forget that, right? We good so far? We're done with the first page. We're done with the front of the first page. Okay. So this is these are actually like real quiz. This was I used to give the quizzes in class, right? But then I put them online because I figured you guys were doing this homework instead. So this is actually like, real questions as you may see them on the exam, perhaps. So let's do some multiple choice questions and we'll keep on going. So we're going to do some mitosis practice, meiosis practice, and also some genetics practice here for these next couple pages. So number 1, let me read through all the options. Which of the following statements about mitosis is false? Letter A, mitosis produces daughter cells with a diploid number of chromosomes. Okay, is that true or false? Okay. Letter B, mitosis produces 2 identical daughter cells. Is that true or false? True. Okay. Letter C, mitosis is preceded by interphase.
That's true.
That is true. Letter D, mitosis is the asexual reproduction of somatic cells. That's true. Letter E, crossing over is a phenomenon that creates genetic diversity during mitosis. That's false, right? So the answer has got to be what? E. Okay, so for the practice little quiz here, for number 1, the correct answer is E. Because crossing over only happens in meiosis. Number 2, which of the following statements about meiosis is false? We'll do this the same way. So, letter A, meiosis produces 2 identical daughter cells?
No.
That's false. So we already got to our answer right there. Number 2, the answer has got to be A, because as you guys just told me, meiosis doesn't produce 2 identical cells. Meiosis produces 4 unique cells. Okay, but let's just read through the rest to confirm that we know that those are all associated with meiosis. So letter B says meiosis produces 4 genetically unique cells, right? There it is. Letter C, meiosis occurs in the sex organs, producing gametes, sperm and eggs. Is that true? Okay. Letter D, there are 2 divisions in meiosis. And letter E, meiosis produces daughter cells with a haploid number of chromosomes. Yes. Okay. So the false one has to be letter A. 2. We are right here. Okay. Are you in there on that class?
No, but I'm in another one.
This is not going to help you if you're in some other
No,
class.
this is not.
I think it's pretty much like the same class.
I just can't guarantee that the questions will be worded the same if you're taking it with another professor.
Thank you.
Okay, so now this one is going to be ID of cells. Okay, so number 3, which cell in the figure below is an interphase? So of these drawings, guys, which of the cells is an interphase? What are you not going to see clearly if the cell is an interphase? You're not going to see what? You're not going to see chromosomes. So in which of these can you not see nice rod-shaped bodies of chromosomes? Picture B. Definitely picture B. So number 3 is letter B. Number 4, which cell in the figure below is in prophase? So the chromosomes have coiled and shortened. Okay.
Is it D?
It is D. You're correct.
Is this mitosis we're talking about?
This is mitosis, yeah.
Okay.
And then for number 5, which cell in the figure below is in metaphase? So chromosomes in the middle of the cell, right?
C.
Letter C. Okay, so that's cell C. Okay, so number 6, all of the offspring of a cross between a red rose and a white rose are pink roses. This means that the allele for red roses is? That's right, incompletely dominant to the allele for white roses. So number 6 is letter D. Okay. Number 7, if big D is dominant to little d, what is the genotype of an individual who is heterozygous for dimples? So which of these genotypes here is a heterozygous genotype? Big D, little d, little d, big D, little d, just big D, or little d, little d, little d? Big D, little d. That's right, choice C. Right? Big D, little d. That's heterozygous because you've got one of each. One dominant allele and one recessive allele. Number 8, we gotta do a Punnett square. I mean, you wouldn't necessarily have to because some of you guys probably know this by now. So let's see, if big A is dominant to little a, what is the expected phenotypic ratio of the cross, big A, little a, crossing with a big A, little a? But just for good practice, we might as well go ahead and do our Punnett square. And so this is the one parent, and so we can set up big A here, little a here. Now the other parent will go big A, little a. So how do we fill in our box? What's going to go here? Big A, big A here. Big A, little a here. Big A, little a here. Little a. Good. So what's our phenotypic ratio? So what does that mean? That means how many individuals are going to express the dominant trait versus the recessive trait? 3 to 1. That's a 3 to 1 phenotypic ratio. So then the answer for number 8 is C, 3 to 1. So the 3 that express the dominant trait are these 3. 3 out of 4 express the dominant trait, and only 1 out of 4 express the recessive trait. Okay. And remember that that's a constant. You will always get that 3 to 1 phenotypic ratio if both parents are heterozygous for one single trait. Okay? All right, so now let's get into a more interesting Punnett square. Number 9. Okay. So number 9, we have rats. And we're talking about the inheritance of fur color. The allele for brown fur is big B, and it is dominant over the allele for white fur, which is little b. So we're going to use a Punnett square to predict the results of a cross between a rat who's homozygous for brown fur and a rat homozygous for white fur. And we're going to look to see what the percentage of offspring can be expected to have that recessive trait white fur. So we got to pull out that important information from that problem, right? So let's look at the genotypes of the parents. So one rat is homozygous for brown fur. What's that rat's genotype? Big B, big B. Homozygous, 2 of the same allele for the dominant color brown. And the other parent is a rat who's homozygous for white fur, the recessive trait. So what's that rat's genotype? Little b. Okay, so now we got to do a Punnett square. And so one parent is big B, so big B. The other parent is little b. So what are we going to put here? Big B, little b. All big B, little b's. So these parents can only make one kind of baby, right? A heterozygous baby with what color fur? Brown. So the question is, what percentage of the offspring can this cross be expected to have white fur? The answer is 0%, right? Letter A, 0%. Because the only way to have white fur is to be little b. There's no little b's here. All heterozygous, all brown. Okay? Okay, question 10 is a monohybrid cross involving blood types. And this is kind of like the hardest blood type question you could get because this has limited information. I know we did a whole lot of practice in that one class on blood types, but this one has limited information. Why am I saying that? What's the limited information?
We don't know if one of the parents is heterozygous or homozygous.
That's exactly right, Dory. So the father, he's blood type A. But there are 2 possible genotypes for blood type A, right? And I didn't tell you that. So you have to do 2 Punnett squares to figure out all possible outcomes if we only have that limited information. So that's what the 2 squares are for. Okay? So father is blood type A. So what 2 possible genotypes would he be? So he could be homozygous for A blood, or he could be AO, heterozygous for A blood. The mother is type AB. So let's go ahead and put an AB on each side of the Punnett square box. Okay, so let's fill it in. What do we get? If the dad is homozygous for A, what do we get? AA, that's A blood. AA. A what? AB. AB. So if he's homozygous for A, he could either be A, they could make A or AB blood babies, right? What if he's heterozygous for A though? What are you gonna get here? AA. What about here? AO. AB, and BO. So what are the blood types possible from this one?
A, B, B.
A. So what's the best answer of your choices? What is it?
B.
You're saying E? Did you say? How could it be E? There's no O E's, right? It's gotta be B. right? You can have A blood babies, B blood babies, and AB blood babies. That's all that's possible. So it's got to be choice B. A and B and AB. Yes. Number 6. Number 6. So a red rose, like from the example from the slides in class. So red is coded for by big R, and white is coded for by big W. So a true breeding red rose would be RR, crossing with a white rose that's big W, big W. And so when they have babies, you get heterozygous offspring, but because these alleles are incompletely dominant to each other, they're pink. So the genotype is all heterozygous RW, but the phenotype is pink. You're not going to have to solve incomplete dominance questions on the test. Okay? Just what you should know is that if you cross a dominant with a recessive, an incomplete dominant, it results in a new phenotype, that tells you it's incomplete dominance.
How does that relate to pleiotropy? Does that have any relationship with pleiotropy?
Pleiotropy means that there's just a gene that affects more than one characteristic.
So like affecting malaria and sickle cell?
Well, so the example with that was the sickle cell disease, and also the allele's ability to offer resistance to sickle cell, to malaria. Okay? So that's pleiotropy. Okay? We can go over that after this, just for the sake of the recording, for the people who are going to be watching from home. I just don't want them to get totally lost in our worksheet here. But we can go over that afterward.
Sure.
Okay, definitely. Okay. All right, so continuing with the worksheet here, so we're done with that page, so now we're going over to this page that says Unit 3 review. Okay, this is from a worksheet, so there's no line heading Unit 3, it's not Unit 3. Just get some room here. Okay. So Mendel's laws. Mendel had many different laws in terms of understanding genetics problems and patterns of inheritance. So number 1, according to the law of independent assortment, which combination of alleles can be produced in a gamete of an individual that has the genotype big A, little a, big B, little b, little c, okay? So which of those could it be? So let's just go through them. Choice A says just big A, little a. Is that possible? In a gamete of an individual who has this genotype? No, because you're missing the B gene and the C gene, right? You need to have one of all 3. What about choice B? It just says big A, big B. Could that be it?
No.
No, because what are you missing?
C.
You're missing the C gene. Okay, so that can't be it. What about choice C? Choice C says big A, big B, big C.
No.
Why can't that? That has one of each, but that's right. In the parent's genotype, there's only little C's. So that parent cannot make a gamete with a big C. So that can't be it. So what about choice D? Does that look right?
Yes.
Choice D, that's where it is. That's the answer. Choice D, because the gamete that could be made from this genotype could be little a, big B, little c. It's got to have one of each allele from each of those genes.
Is there a ratio relative to this?
No, because this is not the outcome of a Punnett square. This will be the result of foiling to figure out the different gamete combinations possible. Number 2, be careful about the wording here. Okay, so this was number 1 on the sheet. This is number 2. The phenotype of an organism is determined by its, what determines the phenotype? The genotype, okay? A lot of you guys may have jumped right to appearance, but if the question was worded a bit differently, the answer could have been appearance. But let me just tell you, if the question said the phenotype of an organism is its, then the answer would be appearance. Okay? But it says the phenotype of an organism is determined by, and so what determines the phenotype? The genotype. Okay, genotype type, choice B. Number 3, if both parents are heterozygous for 2 traits, then their genotype is what? Letter A, big A, little a, big B, little b. What do you think about that? Yeah, you can stop right there. It's choice A. Choice A is big A, little a, big B, little b. Okay? And just as an extra extension question, guys, those 2 parents who have this genotype, when they make babies, what will always be the phenotypic ratio produced by that cross?
9 to 3 to 1.
9 to 3 to 3 to 1. Perfect. Don't forget that, because otherwise you have to do that big dihybrid cross and do all that work to get your answer, but you don't need to do that. If both parents are heterozygous for 2 traits, then you always know that you will get that 9 to 3 to 3 to 1 ratio. 9 out of 16 have both dominant traits, 3 out of 16 have one dominant and one recessive, another 3 out of 16 have one recessive and one dominant, and 1 out of 16 have both recessive traits.
What is D considered? Like little a, B, small b, small
That would be homozygous recessive for 2 traits. Okay, that would be the only genotype that would express both recessive traits. So like our 1 out of 16 would be little a, little b, little b. So what about number 4? In codominance, what's true? Is only one allele expressed? Choice B is neither allele expressed? Choice C, both alleles are expressed?
C.
It's choice C, both alleles are expressed. If it's codominance. What was the example of codominance that we talked about in class?
What type?
Which blood type?
AB?
Oh, it was recessive.
AB?
AB, right? AB is the result of codominance. The A allele for blood type is codominant for the B allele for blood type. And that means that for people who have AB blood, they have both carbohydrates A and B attached to their blood cells. Both alleles are expressed. Both alleles are expressed in codominance. And the example in class was the AB blood type. Okay, you all good right now? Question? What?
So is how many question number 5 or 4?
We were just finishing 4. We're on question 5 now. Number 5, what genotype can a person with blood type B have? Let's just go through those options and eliminate what is obviously not going to be blood type B. Okay? Alright, so blood type B. So for choice A, we have 2 undercase I's. What blood type does that indicate? That indicates O blood, right? So it can't be that. So choice A is I, little I. That's O blood. So that's not it. What about choice B? Choice B is AB. What blood type is that?
AB.
That's AB blood, so that can't be it. Choice C just gives you one allele. Is that a blood type? Is that a genotype?
So what I put in none?
We didn't finish all the options yet. Okay. Choice C, right? You only have one allele. You need 2 alleles for a genotype. So choice C is not a real blood type. What about choice D? What's that? The genotype given by choice D is BO, because the undercase I means O. So it's that. It's choice D. Right? That's BO. To have B blood, you could be BB or BO. Okay, remember that on my paper exams, you won't see those I's. I'll just use the letters. determine what? A, the cause of the disease. B, how a trait is passed from one generation to the next. Letter C, which member of a family will definitely get a particular trait. Or choice D, which chromosome carries the allele for a particular trait.
B.
Yeah, choice B, how a trait is passed from one generation to the next.
Is that the same thing as heredity?
Yeah, you think that pedigree traces the inheritance or the heredity of specific traits. Yeah, you can say that. Okay, and then number 7 we're just omitting, but number 8. Why are X-linked conditions such as hemophilia much more common in males than females?
Only have one X.
That's right. Because males only get one X chromosome, right? And if they have that recessive allele linked to that single X chromosome, they've got the trait. Okay? Males only need one affected X chromosome to have the trait. So an example of a genotype of a male who has hemophilia is what? X. What? Big H? Little h. Y. Okay. So this would be the genotype of a male who has hemophilia. For a female to have hemophilia, she would have to be what? X little h. No big h's. Little h's. The female would have to inherit 2 affected X chromosomes, one from mom and one from dad. Okay? So it's possible for a female to get hereditary hemophilia, but it's much less likely because she has to inherit it from both parents. The dad only has to inherit it from his mom to get it. Okay? So males only need one affected X chromosome to have the trait. Females would need 2. Okay, does that make sense? We did a lot of genetics practice in class, and that's why we didn't do as much here. We're going to save the rest of the stuff that we just talked about the however long ago, almost a week and a half ago, right? Chapter 10 stuff on DNA replication, transcription, and translation. That's what we're going to spend the remainder of our time with here today with these last couple pages. And remember, if you have any other questions, you can always stay behind after we get finished with the worksheet. We'll go over some things in more depth. Okay, so let's start over here. So we're on to the Chapter 10, concepts of DNA to RNA to protein, right? That's the whole purpose of Chapter 10 is to go over the central dogma of the flow of genetic information in cells, from DNA to RNA to protein. So let's first talk about DNA a little bit. So, number 1, the structure of DNA was determined by who? Watson and Crick.
Professor,
Yes.
sorry for interrupting.
No problem.
Which page are we?
Excuse me?
Which page are
We are on the page that has at the top Chapter 10 review.
Perfect, thank you very much.
But did that actually come from Rosalind Franklin?
Well, they used her work to eventually be credited with the discovery. But, so what you want to know about Rosalind Franklin is that she did X-ray crystallography to take a snapshot of DNA, but then she died. And so then Crick took her work and joined together with Watson, and they continued it, and they were the ones who eventually were credited with the discovery of the structure of DNA. Okay? So Watson and Crick, and what did they do? They described the shape of the DNA molecule as a double helix. Okay, so that's all the answers are number 1. So the structure of DNA was determined by Watson and Crick. who described the shape of the DNA molecule as a double helix. Number 2, the double helix is held together by what kind of bonds? Hydrogen bonds that form between the nitrogen bases. After DNA replication, how many identical molecules of DNA are produced? 2 identical molecules of DNA. All right, now we're going to summarize the processes of transcription and translation. So we want to say, where do they occur and what happens? So number 4, transcription versus translation. Okay, so where does it happen and what happens? All right. So where does transcription happen?
Nucleus.
In the nucleus. And what happens there in the nucleus during transcription?
DNA is transcribed into RNA.
Okay, yeah, so DNA is transcribed into RNA. And so to use words other than transcribed, okay, it's not improper what you said, we can say that the nucleotide language of DNA is converted to the nucleotide language of RNA, right? Okay, so we can say DNA gets converted to RNA in transcription. But specifically what's going on is that the nucleotide language of DNA, which is what? What's the nucleotide language of DNA?
ACTG.
Yeah, A, T, C, G is being converted to the nucleotide language of RNA, which is what? A, U, C, G. A, U, C, G. Good. Okay. We'll give you some examples of that shortly. And then in translation, where does that happen? Where does translation occur? The cytoplasm. And what happens during translation?
Codons pull up,
So RNA codons are converted into amino acids, which are the building blocks of proteins. Okay? So what we have here is RNA being converted into proteins. That's the general summary. Okay? But specifically what's happening is that RNA codons, which is a set of 3 RNA nucleotides, are coding for amino acids, which link together to build proteins. RNA codons code for amino acids. And then amino acids get bonded together to build a protein chain. Okay, and we're going to do examples of that shortly. Number 5, though, let's go ahead and do number 5. What types of mutations can occur during replication or transcription, and what factors can cause them? So, what are some types? Types of mutations. And these are not cases of inheritance, okay? I don't want things like Down syndrome. That's because of nondisjunction. What are errors that happen during replication or transcription called? There are deletions is one. What else?
Insertions.
Insertions. What else? What's the one that only results in the change of one codon? These shift the whole reading frame and change every codon down the line. But which one is the result of only changing one codon? Remember that one? No? Drug like that. How about base substitutions? Remember that one? These are the types of mutations that can occur. And what are the causes of these mutations? There are two general causes. What are the causes? What can...
Spontaneous.
Yeah, they could be spontaneous. So that just means random. So like it could just happen to anybody randomly. It's just a random error, a random mistake. Or it could be exposure to what kind of things in our environment? Mutagens, or exposure to mutagens. And what are examples of mutagens? Radiation. And what else? Chemicals. Did your mother ever tell you not to stand in front of the microwave when it's on? Yeah, maybe not. That's because you don't want to be exposed to that radiation. Okay? Radiation and chemicals are mutagens that can cause an error in these processes. Okay? Alright, let's get to the serious business and practice some replication and then some transcription here. Okay, so for number 6, it says after DNA replication, what is the corresponding strand to pair up with? And I have here a parental DNA strand. So here, this is our original strand of DNA, a little segment of DNA, really. And the sequence of nitrogen bases is T, G, G, A, C, A. Okay? And so we're going to make a new strand of DNA. So what is the enzyme DNA polymerase going to put here to match up with the template? What's our nucleotide language of DNA? So let's review. What bonds with what in DNA? A bonds with T and C bonds with G. Okay, so if you have a T in the original DNA, what are you going to put? A. If you've got a G in the original DNA, what are you going to do? C. Add C. We've got a G?
C.
We add a C. We've got an A?
T.
We add a T. We've got a C?
G.
We add a G, and if we've got an A?
T.
We add a T. Okay? So our new segment of DNA that matches our template is ACC. TGT. To come up with that, all you have to do is remember those ATCG pairing rules. Okay? So that was number 6. That's our replication. Number 7, we're not doing replication, we're doing transcription. So we're going to make RNA out of DNA. So we have our parental DNA segments, and we're starting off with the same sequence. T, G, G, A, C, A.
This could be A bonds with U.
Hold on a second. So our new RNA strand after transcription. So just remember, so wait, what are the RNA nucleotides? Let's review them. What are they? They're still here, but let's say them. A, U, C, G, okay? So basically, anywhere that you would have normally put a T, there is no T in RNA. So instead of putting T, you're going to put U, okay? Because the RNA to RNA base pairing rules are A bonds with U, C bonds with G. Okay? So what are we going to put? What's going to bond with the T in the DNA?
A.
A. A. Okay.
There's already a T there, so that's what it's getting.
Instead of T, put U. You wouldn't put a T here, right? You would put A here. Are you going to put anything different here? You wouldn't put a T here if this was a DNA replication, right? The T was already here in the DNA. You gotta do purines with pyrimidines, remember that? Okay? So what are you gonna do here? What's gonna bond with this T in the RNA? A. What's What's in the bottom of the G of the DNA? C. What's in the bottom of G?
C.
What's in the bottom of A in the DNA? U. There's the U.
G.
What's in the bottom of C?
G.
G. What's in the bottom of A? U. If you're starting off with T in DNA, you're still going to put A. Okay? But if you're starting off with A in the DNA, you gotta put U, because there's no T. There's no T in DNA. Okay? So the new sequence is ACCUGQ. That's our signature of RNA after transcription. So you see how when we summarize this here, that what we're going is the nucleotide language of DNA getting converted into the nucleotide language of RNA. Okay? And where does this happen? This part, this time? Nucleus, right? Nucleus. Transcription we just did. We didn't do translation, we just did transcription. Okay? Alright, so be sure to know how to do that if you're given a segment of DNA, either replicate it to make new DNA or transcribe it to make complementary RNA. Okay,
You
so
said it
let's,
was transcription, right?
what is it?
It was transcription.
This is transcription, and this is replication. Number 6 was replication, number 7 was transcription. Okay, so next worksheet, this one says Unit 4 review. Let's see what this worksheet is. So number 1, which represents a correct base pair found in a DNA molecule? This is a really easy question. Okay, what do you say? It's got to be choice C, right? C bonds with G. Right? Choice C, C bonds with G. Because if you look at the other options, right? Choice A, does G bond with A? No. Choice B, does C bond with T? No. Okay. And choice D, does T bond with U? No. Okay, so the correct answer can only be C bonds with G. Right? In DNA, A bonds with T, C bonds with G. Number 2, DNA replication involves what? Let's go through each of them. Number A, the synthesis of proteins? No. Choice B, the formation of messenger RNA?
Yes.
Are you sure? Replication?
Yes.
Replication of DNA? No. What results in the formation of messenger RNA? What process does that?
Translation.
Transcription. Okay, if the question said producing RNA from DNA, you know, if it said transcription involves, you would say formation of messenger RNA. But formation of messenger RNA happens during transcription. What about letter C? Separation of the 2 strands that make up the double helix. Yes, definitely, right? Because when DNA is going to replicate, it has to unwind to form those bubbles, right? So choice C definitely works. Let's look at choice D. Substitution of the nitrogen-containing base T in the place of G? No, that's a mutation. Forget about that. So number 2 has got to be choice C. Number 3, unlike DNA, RNA, letter A, is a large molecule made up of nucleotides. Is that the difference between DNA and RNA? No, so that can't be the answer. Unlike DNA, RNA contains the sugar ribose. Yes, that's true, right? And that's the difference between DNA and RNA, right? Because what sugar does DNA have? Deoxyribose. So the correct answer for number 3 has to be letter B, contains the sugar ribose. Because the other ones, so letter C says is found mainly in the nucleus of the cell. That's true. And then letter D says contains the nitrogen-containing base thymine. DNA has thymine, right? But RNA doesn't, so it's got to be choice B. RNA contains the sugar ribose. DNA does not. Number 4, what is true about transcription? Now be careful of the language here. RNA is synthesized from DNA. That means RNA is made from DNA, okay? Does that sound right?
Yeah.
RNA
So, yeah.
Does that
RNA is synthesized from, made from DNA.
does that mean DNA is transcribed into RNA?
Yes. But that's not using the word transcribe. That's the whole thing. What does that mean? Okay? What happens in transcription? DNA is converted into RNA. So this just says that backwards. RNA is synthesized from DNA. RNA is made from DNA. So it's going to be choice A, right? Because the other one says, choice B says DNA is made from RNA. That's not true. That's not what happens. Letter C says a protein is made up of ribosomes. That's not what happens. That happens after the end of translation. After translation's done. And letter D, an exact copy of a DNA molecule is made in the nucleus. What process results in that? Starts with an R.
That's replication.
That's replication. That's replication, choice D. So the only answer that could be right here is choice A. RNA is synthesized from, or is made from, DNA. I love this one, codon-anticodon pairing. Hooray. So in translation, what anticodon on tRNA pairs with the mRNA codon AUC? So we're using mRNA base pairing rules, which is what? A bonds with U and
CG.
C bonds with G. So let's write this up. So our mRNA codon is AUC. So to figure out what our tRNA anticodon is, what are we going to do? Without even looking at those options, figure it out first. Okay? So what's going to bond with A? U. What's going to bond with U? A. What's going to bond with C? G. Do you see that answer, UAG, as one of your choices in multiple choice?
Yes.
Yes. Choice C. Choice C. UAG. Okay, you just got to do RNA to RNA base pair rules. AU, UG. Number 6. Amino acids are assembled into a specific order. Let's see what's true. On the ribosome, is that where it happens? What? Remember what the ribosomes are back from chapter 4? They're the site of protein synthesis. So that means amino acids are being linked together to build a protein. So are amino acids assembled in a specific order on ribosomes? Yes. Are amino acids assembled into a specific order according to the sequence of nitrogen-containing bases, i.e. codons? Yes. Is it done depending on codon-anticodon pairings?
Yeah.
What brings them? What brings the amino acids to the ribosomes? tRNA, because of this pairing. So the correct answer is all of the above. All of the above are correct. Number 7, what is meant by a double helix? Good question.
Like a twisted ladder or
Okay. So what?
stranded
So we got 2 strands of what?
DNA.
Well, what is DNA? That's what it's saying. What is meant by a double helix? So 2 strands of polynucleotides wrapped around each other. 2 strands of polynucleotides. Nucleotides are the building block of DNA. So like one sugar group, one phosphate group, one nitrogen base makes it a nucleotide. But you've got like millions of those all linked together. And then you've got 2 separate strands of them all wrapped around each other. And that makes up your double helix.
It has to be poly, that's more than one.
That's right, because it's many nucleotides all wrapped around each other. So like a simple drawing of DNA would look something like this. That's your 2 strands. Okay. Along the strands are alternating groups of sugar and phosphate. So sugar, the black is phosphate, sugar, black is phosphate. So you do sugar, phosphate, all the way up and down each strand. You can just keep on going all the way up and down each strand. And then extending from the backbone, the strands are what? The nitrogen bases, right? Like what? What are the nitrogen bases, guys? We talk a lot about them. Well, we're talking about DNA, so ATCG, right? So then these are the nitrogen bases, so we can say this is A, this is T, and so this is T, and this is A, and this is G, and this is C. This is G, and this is C. Okay, so, that's the general structure of a DNA double helix. 2 strands of polynucleotides consisting of sugar, phosphate, nitrogen base, wrapped around each other. That makes our double helix. And it can be described as a twisted rope ladder. And just for further review, guys, what holds together the nitrogen bases? Hydrogen bonds. Hydrogen bonds, right? We had already talked about that on one of the previous pages. Hydrogen bonds hold the nitrogen bases together, and the 2 strands as a whole, causing that double helix shape. Okay? And number 8, what is the role of ribosomes in protein synthesis? The ribosomes are where what is brought?
The site of protein.
That's right, it's the site of protein synthesis. So the ribosomes are the site of protein synthesis. And to get rid of those words protein synthesis, because that's in the question, you can say this is where amino acids are linked to build a protein. Yes, it's true. Ribosomes are the site of protein synthesis. What does that mean? Amino acids are being brought there by tRNA, and then our RNA is helping to link them together via a peptide bond to make a protein. Remember that? One more time. tRNA picks up and brings those amino acids to the ribosomes, the site of protein synthesis, where then that other type of RNA, ribosomal RNA, rRNA, helps to link them together, right? And what specific bond links together amino acids? Peptide bonds. You guys learned that a long time ago, back in the beginning of the term. Okay, don't forget it. See, we're bringing it all together, these larger concepts, okay, to make a big protein chain. Okay, last page, front and back to do. Okay, so one more time, we're going to keep on reviewing about transcription here, and then in the back of the page, we're going to do it all. We're going to do transcription and translation, and actually using the codon table. It's going to be really cool. Huh? We're on the worksheet now that says RNA and transcription. It's a big fill in the blank activity. That's where we're at. Okay, so I'm going to just read through the paragraph, guys, and you guys shout out the answer as we get to it, and I'll write them on the board. Okay. Okay, so RNA is different from DNA in several ways. Whereas DNA contains the sugar, deoxyribose, RNA contains the sugar, ribose. In addition, RNA is made using the nitrogen-containing base, uracil, while DNA contains the base, thymine. RNA and DNA, however, contain what kind of groups?
Phosphate.
Yeah, phosphate groups. Phosphate groups. Okay, unlike DNA, RNA exists in 3 different types. One type known as messenger RNA carries the genetic information. Messenger RNA carries the genetic information from the nucleus to the cytoplasm, right? That's why it's called messenger RNA, because it carries the message, the genetic message, out of the nucleus into the cytoplasm for translation, eventually. Okay? And that's, so it carries the genetic information from the nucleus to the cytoplasm, where then the proteins are synthesized, right, during translation. A second type of RNA, which is called what? Transfer RNA. Transports the amino acids to the ribosomes. And then next sentence, there's a little bit of a typo here in number 2. It should say these structures make proteins. These structures make proteins. Okay, so number 10 is ribosomes. These structures make proteins. Okay, the third type of RNA is called ribosomal RNA. All 3 types of RNA are made from DNA in a process called transcription. Just as the DNA does in replication, the 2 strands of the double helix separate. Then DNA makes a strand of RNA that is complementary to the base sequence. Complementary to the base sequence of the DNA. Therefore, the nitrogen-containing base guanine will pair with the cytosine base in DNA, while this will pair with the thymine base in DNA. Adenine. Okay, and we didn't use one of these words. So what word did we not use? Cytosine. Cytosine is a little DNA and RNA, which is we didn't use it in that passage. Okay, so that's all right, the grand finale. And I usually like to do this in class after the chapter 10 lecture, but we had gone over the genetics problems at the beginning of class. We didn't have any time, so we're going to do it now. Hooray! And this really kind of helps to do it. Does anybody think that they have done it right? Is that all you do? Okay, cool. All right, so let's go ahead. I gotta erase all this. Do you guys have these answers down? So I gotta write this whole sequence on the board. So we're gonna do transcription and translation here with this big giant sequence of DNA. Now on a test, you wouldn't have nearly as giant of a sequence. It would be a bit smaller, but it could be 12 long or so. Most commonly it's like 6 long. But that's it. It's a big one. You guys have all this down? Yes. Okay. I'll start with what I started with. Just bear with me while I break down the big sequence, and we'll start transcribing and translating. So that big sequence there, you see, that is a strand of DNA, right? Because when we do transcription, only one side of the DNA strand is used as a template for transcription. So let's see, our DNA is G, C. Just make sure that you guys make sure that I don't omit one here. T, A, C, A, T, G, T, C. Did I copy that right? Did I mess up somewhere? If I messed up somewhere, I've got deletions and insertions, and I mess up everything. So, okay. All right, so that's our DNA. So for number 1, it says we're going to transcribe this sequence into RNA. So we have to do our transcription rules here, right? So A bonds with C, and maybe A bonds with U, sorry, C bonds with G, right? Anywhere where you would normally put T, you're going to put U instead. Okay, so let me go ahead and change my color here. Black. Let me change this to black. RNA, okay. So let me hear it, guys. Go ahead and give me the answer here. So let's go. C, 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 G, G,
G,
G,
A, U, A,
C,
G,
So we got G, yeah. U, A,
C,
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 G,
G,
C. All right, guys are all out of sync. Okay. We've got A in DNA next. What are we going to put? U. You got A. Another U. C. A. G.
U, G, A, U, G.
Okay, good. Transcription done. Where did that happen? The nucleus. Okay? The nucleotide language of DNA got converted to the nucleotide language of RNA. Now's the fun part. Now we get to do translation. So in translation, what do we have to do? What initiates translation? The reading of what special codon? AUG. Okay, so for translation, okay, so we're going to get amino acids here, which eventually build proteins, right? So we're going to read until we find our start codon, right? So we're going to go, no start codon. Here it is, right? Start. Translation, right? Initiation. Initiate translation. All right, so we got our first codon, it's our start codon. So now we divide out the rest into 3. So UAC, that's our next codon. Look at the table. What does UAC code for? What amino acid? Do you guys see it? UAC, tyrosine in the chart. You would never have to memorize this chart. You would be provided it. Okay? The next codon is CGG. So what amino acid is that? CGG. Arginine. Next codon, UUU. Phenylalanine. And by the way, phenylalanine is something that's added to like all gums today. Okay, I guarantee you, if you have a pack of gum on you, look at it. It says it has phenylalanine added to it for whatever reason. Phenylalanine. The next codon is CCA. What amino acid do we get from CCA? Proline. Next codon, GUG, what do we get? What is it, Valine. Valine. You guys see that? Valine, GUG. And AAU, what's that? Asparagine. And then UAA, what's that? Stop. Termination of translation, right? Initiation. Elongation. Add an amino That's translation. Initiation, elongation, and then termination.
And what's that extra G again?
That's a non-coding region. It's just non-coded. It's just not used. Same thing with this part in the beginning, non-coding. Interestingly, most human DNA that eventually gets converted into RNA is non-coding. Only about 1.5% of all of the information that was in our DNA actually becomes coding for traits. The rest is non-coding. What is it, Tori?
How do we know if it's not coding?
It's not in between start and stop codons. It won't be coding if it's not in between start and stop codons. Isn't that cool? So that's transcription and translation.